So our next question was, what are the important sequences in a promoter? So we spent a long time really find this area that RNA polymerase has to bind to and open up to start the transcription. But we also know that promoters do more than just recruit RNA polymerase. It has to recruit these regions also in response to environmental or growth cues, for example. So are there other regions that are important for that? And so this took advantage of a yeast gene where we have a full coding region, so that's the starting from the transcriptional start site and what actually makes the protein. And then there was this region upstream that they knew was the promoter, or was important for transcription. And so the real question was it this entire region that was important for transcription, or were there regions that were more or less important? And so from this gene, the yeast, there was two different phenotypes that they were measuring. The yeast could have doubling time, so their growth rate, that's like what we saw with the bacteria with mesalcin and stall. And then also they could measure the drug resistance. And so these were two different phenotypes. And when you have the full promoter, so that full 320 base pairs, then the yeast grew in 3 hours and they were drug resistant. On the other end of the spectrum, if there was no promoter, basically just 29 base pairs upstream, the yeast did not grow, and so then they were also not drug resistant. So we know that there is important sequences in there between the 300 base pairs and 29 base pairs. Now, the researchers made slightly bigger and bigger constructs to try to map what are the important activator domains. So they took a construct that was 78 base pairs upstream, and they saw, now the yeast are starting to be able to grow, so they have a doubling time of 5 hours. So we have some important activator domain that we have put back in to get this gene transcribed, but it's the yeast are still not drug resistant, so that can't be the whole story. They added in another 113 base pairs, so about 50 more base pairs upstream than they were looking at the 78, and we saw again that there was the 5 hours that we had seen before, so the doubling time was same, but we also still have no drug resistance, so we didn't really add any other information between that 113 and 78 base pairs. They made another construct that was 155 base pairs upstream, and now this time it both has we're seeing a growth rate. The yeast are able to grow again at 5 hours, and the drug resistance was restored. So now the yeast were resistant to that drug. So from this experiment, we can conclude that there was two different activator domains, one in the region from 78 base pairs to 29 base pairs. That likely includes the things that we've seen before, the ta-ta box, the real nuts and bolts to getting transcription going. But then there was another activator domain that also confirmed this drug resistant upstream. And another thing that's interesting to note is that these two activator domains are not contiguous. They are not right next to each other, so that gets you starting to think, okay, how can we have the architecture of a gene and the different parts of a promoter?